This video will demonstrate the procedures used in DNA Ploidy analysis. Referring to the tumor area marked previously by a pathologist on an H&E stained slide, the corresponding tumor area of the paraffin-embedded tissue is outlined with a needle. The block is then cut in 50 micrometer thick sections. Preparation of the tissue sections begins with 15 minutes of deparaffinization using xylene. This is done twice. After discarding the xylene, the tissue sections are washed and rehydrated with a series of decreasing ethanol concentration: absolute, 96%, 70%, and 50%. During the rehydration process, the tissue sections are transferred to conical centrifugal tubes. After rehydration, the tissue is washed using phosphate-buffered saline (PBS), and the test tubes are spun in a centrifuge at 200 G for 10 minutes. The supernatant is then removed with a pipette, and new PBS is added before a second spinning. Here we see the liberation of single nuclei through enzymatic digestion and mechanical disruption of the tissue. Fresh enzymatic solution is made by dissolving proteinase type 8 in PBS, and mechanical disruption is performed by magnetic stirring bars. 1 milliliter of enzyme solution and 1 magnetic stirring bar are added to the tissue and placed on a magnetic stirrer. Tissue disruption is performed at room temperature for 60 to 75 minutes, depending on the tissue type. The enzymatic reaction is stopped after tissue disruption by diluting enzymatic solution 7 times with cold PBS, and the magnetic stirring bars are removed. In order to filter tissue homogenate and make the nuclei solution as pure as possible, it is filtered through a 60 micrometer nylon mesh. The filtrate is then centrifuged at 800 G for 20 minutes to pellet the nuclei. Then, the supernatant is discarded, and 0.5 to 3 milliliters of fresh PBS is added depending on the size of the pellet. Cytospin funnel, filter, and polyolefin-coated slide are assembled for the making of a monolayer. 100 to 500 micrometer of nuclear solution suspension is added to the funnel, depending on its concentration. Samples are spun for 5 minutes with a speed of 40 G in a cytospin. After cytospinning, the density of nuclei is inspected under a microscope. The monolayers should contain a significant amount of nuclei to ensure fast measurement and proper classification. Finished monolayers are dried and fixed in formalin overnight before proceeding with the Feulgen staining. The Feulgen staining process begins with hydrolysis using 5 molar of hydrochloric acid. The slides are left to bathe for 60 minutes at room temperature. During this process, DNA produces free aldehyde groups which can be detected by a color reaction. Subsequently, the free aldehyde groups are stained using the Schiff reagent. This is done for 2 hours, under dark conditions. The Schiff's reagent binds to the aldehyde groups in DNA, which results in a magenta color compound. Following staining, the monolayers are bathed in a 1 molar sodium sulfate solution. This is done 3 times for 10 minutes. The monolayers are then dehydrated with a series of increasing concentrations of ethanol: 70%, 96%, absolute, and absolute. After dehydration, the slides are dipped in concentrated xylene. Cover slips are applied to the slides. The monolayer is now prepared for digital imaging. The DNA content in the nuclei is measured with the Ploidy Workstation (PWS) DNA analysis software developed by ICGI. PWS consists of two applications, PWS Grabber and PWS Classifier. PWS Grabber steers a motorized microscope which is fitted with a high-resolution digital camera. This apparatus scans the slide by incrementally traversing the surface area of the sample until the whole monolayer has been digitally imaged. Up to 900 images are captured for each slide. PWS Grabber algorithms automatically assign nuclei candidates to one of 5 categories. Epithelial nuclei, lymphocytes, plasma cells, stromal cells, and exempt nuclei. These groups are sorted into galleries designed to be viewed in PWS Classifier. PWS Classifier is used to manually verify the accuracy of the Grabber algorithms and ensure each nucleus is complete, uncut, and not overlapping. Once the dataset is determined fit, the integrated optical density, IOD, is calculated by summing up the optical density of each pixel in the nucleus. The IOD value and total nuclei count are plotted in a histogram, which is analyzed and classified into one of 4 categories: diploid, tetraploid, polyploid, or aneuploid. The attending physician is informed of the result.